We're not talking about section 3 for chapter 12 on nervous tissue and introduction to the nervous system. We're talking about synapses. For the objectives you should be able to glean from this section, we should be able to define a synapse and describe the essential structural and functional differences between a chemical synapse and an electrical synapse. It's a fairly short segment, but synapses are important. And so, first of all, a synapse is a place where the neuron connects to another neuron or an effector. There are two types. There are chemical and there are electrical. The chemical synapses are far more common than the electrical synapses. An electrical synapse has presynaptic and postsynaptic neurons that are bound together by gap junctions. And electrical signals travel on the gap, and they can move much faster than a chemical messenger would, or that a chemical synapse would function. It's very fast. There's no synaptic delay in passing along an electrical signal. Chemical synapse, the presynaptic neuron's axon terminal produces this signal. The postsynaptic neuron receives the signal. It's most commonly with one of its dendrites. Synaptic cleft is that small fluid-filled gap between the two neurons, and the events of that synaptic communication, the neurotransmitter molecules are released from the vesicles synaptic knob, very similar when we're talking about muscular activation as well, electrical signal or action potential with an electrical signal for a muscle. In this case, from the synaptic knob into the cleft, and then that neurotransmitter diffuses across the cleft and binds to postsynaptic receptors. And in that binding of the neurotransmitter then to the receptor initiates the postsynaptic potential, a graded potential. And the synaptic delay is the time it takes for all of these events. So you should be able to note what a chemical synapse is within the nervous system and contrast it to an electrical synapse and how does it function. With our fourth section here, we're talking about glial cells. They are primary support with our neurons. You should be able to, with this section, to list the distinguishing features of glial cells, describe the structure and the function of the 4 types of glial cells within the CNS, and the 2 types of glial cells within the PNS. So we have 4 and 2, CNS and PNS, for the types of glial cells. You should be able to define myelination and describe the composition and function of a myelin sheath, distinguish between the myelination process carried out by neurolemmacytes in the PNS and by oligodendrocytes in the CNS. So glial cells, there are neuroglia, also referred to that way, are non-excitable support cells found in both the CNS and the PNS. They're smaller, but they far outnumber the neurons. They account for about half the volume of the nervous system. They're capable of mitosis and dividing and creating new glial cells. They protect and nourish neurons. They provide physical scaffolding for the nervous tissue. They guide migrating neurons during development. They're critical for normal function at neural synapses. The types of glial cells. Astrocytes are star-shaped cells. They have processes that end in perivascular feet. They're the most abundant glial cell in the CNS. They help form the blood-brain barrier by wrapping feet around the brain capillaries. They control which substances have access to the brain. They regulate tissue fluid composition, the chemical environment around the neurons. They can regulate potassium concentration, which is a key component. They form structural support for nearby neurons. They assist neuronal development, and they occupy the space of dying neurons. The second of our glial type cells for the CNS, the epidermal cells, basically line our cavities of our brain and our spinal cord. They're part of the choroid plexus, which produces the cerebrospinal fluid. The microglia are the small cells that wander the CNS and replicate in infection. They're phagocytic cells of the immune system, and they engulf infectious agents and remove debris. The oligodendrocytes are the large cells with slender extensions. They have extensions that wrap around axons of neurons, forming a myelin sheath. And here we have a little closer look at our 4 types of glial cells within the CNS. Let's go here, our image first here. So you have an astrocyte, this big guy here, the perivascular feet. There's a capillary, ependymal cell here. Here's the ventricle of the brain on the outside here. Oligodendrocyte here with lots of unusual branching shapes here. The myelinated axon here, the oligodendrocyte, as being a neuron here. Let's see, and then there we go. There's our microglial cell notification there for our microglial cell here. And of course, remember all oligodendrocytes here, the big guys here, the guys that looks like they have quite a few unusual shaped branches coming out here. They myelinate, insulate our CNS axons, and they allow faster action potential propagation along the axons within the CNS, those oligodendrocytes. And those astrocytes, these big green guy here, helps form the blood-brain barrier. It regulates the interstitial fluid composition outside of our cells. It provides a structural support and organization of the central nervous system and assists with neuronal development and replicates to occupy space of dying neurons. That's why we often hear about them the most. The ependymal cell functions here, these guys, along with each other here, they line the ventricles of our brain, as it shows here, and they helps form the central canal, or along, I should say, the central canal of our spinal cord. They assist in the production and circulation of cerebrospinal fluid. Once again, and our microglial cells, these little guys are those phagocytic cells that move through the CNS. They protect the CNS by engulfing those infectious agents and other potentially harmful substances. So our types of glial cells, then our PNS now, they're just two, our satellite cells and our neurolemmacytes, like our Schwann cells, just like we have Schwann cells for our muscles, and our neurons for our muscles, our skeletal muscles, we talk about as well. And with our satellite cells here, they're arranged around the neuronal cell bodies in a ganglion. They electrically insulate and regulate the exchange of nutrients in the wastes. And the neurolemmacytes, the Schwann cells, they're elongated flat cells that ensheath the PNS axons with myelin, and they allow for faster action potential propagation. So here we can see our two types of cells in our PNS for our neuronal cells. Look at our image here first. There's our satellite cell, and there's our neurolemmacyte. And again, that satellite cell electrically insulates the PNS cell bodies, and it regulates nutrient and waste exchange for cell bodies in the ganglia. And that neurolemmacyte function helps myelinate, as you're seeing here, the myelination around that axonal body, and insulates the PNS axons. It allows for a much faster action potential propagation along the axon. When you get the myelination in general, what we're talking about when myelination occurs, there's a much faster propagation of electrical signals, so information travels faster, which means you get a faster response, technically then an more efficient and potentially more effective response, which is also what happens when we're talking about muscle function, let alone function of cells in general. And when I put something in red for you, it's that it's like all things, none of the information here is completely unimportant, but it's just not a primary focus for us. And rather than just deleting the slide out, understanding tumors, the central nervous system is something of importance. Just for now, it's something we're going to forego. Myelination is the process of the wrapping of an axon with myelin. And that myelin are several layers of membrane of glial cells. And it's a high lipid content, which gives it its glossy white appearance and insulates that axon. And the glia are neurolemmacytes within the PNS, and they're oligodendrocytes in the CNS. So within that PNS, those neurolemmacytes encircle the neuron axon and wrap it in the layers, forming a myelin sheath. And those neurolemmacytes, cytoplasm and nucleus, are pushed to the periphery, forming the neurolemma. And the neurolemmacyte can myelinate only about 1 millimeter of an axon, so several are needed for one axon. Okay? And the gaps between those neurolemmacytes are the neurofibril nodes, also known as the nodes of Ranvier. So here we have our myelination, our PNS axons. So we have our neurolemmacyte that starts to wrap around 1 millimeter portion, the neurolemmacyte here, there's the nucleus, and basically here's our wrapping in this direction around the axon. And then the neurolemmacyte, cytoplasm, and the plasma membrane begin to form consecutive layers around that axon as the wrapping continues. And the overlapping inner layers, that the neurolemmacyte plasma membrane helps form that myelin sheath. See that layering here, that great myelin sheath. The point where eventually that neurolemmacyte, cytoplasm, and nucleus are pushed to the periphery of the cell to form the neurolemma. Okay, here's our myelin sheath. And basically the neurolemmacytes on the outside here, that neurolemma, okay? And that's gonna help that much faster propagation, much more efficient communication. And the myelination by neurolemmacytes within that PNS, you can see the neuron cell body, the neurolemmacytes here, our neurofibril node, there's our axon, that neurolemma, and that myelin sheath continuing to build around the outside of that axon. For the myelination of our oligodendrocytes, and the CNS, one oligodendrocyte can myelinate 1 millimeter of multiple axons, each at multiple spots. There's no neurolemma formed. Those neurofibril nodes here within, between adjacent wrap segments. Are unmyelinated axons in the PNS. The axon sits in the depressed portion of the neurolemmacyte, okay, over here, and the neurolemmacyte starts to envelop those multiple axons. And the non-myelinated axons are enveloped by the neurolemmacyte, but there are no myelin sheath layers around each axon, the unmyelinated axons that are not associated with oligodendrocytes in the CNS. With multiple sclerosis, Very briefly here, it's a progressive demyelination of the neurons to the CNS, so it slows our response, what really is happening. It's autoimmune disorder. The oligodendrocytes within the CNS are attacked by immune cells, so our oligodendrocytes we want are being attacked by our own immune system, which is what happens in autoimmune disorders in general. Body doesn't recognize its own functioning characteristics or functioning components, and it starts to attack itself. So you have repeated inflammatory events that cause scarring and permanent loss of function eventually. The Guillain-Barré syndrome, loss of myelin from peripheral nerves due to inflammation. There's muscle weakness that begins in distal limbs and advances to the proximal muscles. Most functions recovered with Guillain-Barré with little medication intervention. So what did you learn? If a person has a brain tumor, is it more likely to have developed from neurons or from glial cells? Why? If a person suffers from meningitis and inflammation of the meningeal coverings around the brain, which type of glial cell usually replicates in response to the infection? Which specific type of glial cell forms a myelin sheath associated with axons in the PNS? And what's the function of the myelin sheath? How does myelination of axons occur in the PNS? And our final little section here for the basic structure and generalized function, then we have much deeper components to overall function in our final sections here for chapter 12. But here we're talking about axon regeneration. You should be able to identify the factors that influence regeneration of PNS axons and explain why axon regeneration in the CNS is limited. Describe the events of Wallerian degeneration and axon regrowth. After traumatic injuries, the PNS axons can regenerate. It's possible however, if the neuron cell body is intact and if enough neurolemma remains. Regeneration success is more likely if the amount of damage is less extensive and there's distance between the site of damage and structure innervates is shorter. The steps of the axon regeneration, you don't have to know them by rote. General understanding is important here, but you're not going to be tested on the different steps in axon regeneration. But the axon is severed by the trauma. It's proximal to the cut. The axon seals off and swells. Distal to the cut, the axon and sheath degenerate. That's Wallerian degeneration, which you won't technically have to know for your exam. I'll tell you in advance. But that's at a later time that may become more and more vital, not for now. But the neurolemma survives. The neurolemma and the neurium form a regeneration tube. And the axon regenerates guided by nerve growth factors that are released by the neurolemmacytes. That axon reinnervates the original effector or the sensory receptor. And so in that respect, I'm not going to go over the individual steps here once again for the regeneration of the PNS axons. But CNS axon regeneration is extremely limited. The oligodendrocytes secrete growth-inhibiting molecules, not growth factors. A large number of axons crowd the CNS, and the regrowth is obstructed by scars from astrocytes and connective tissue. So with this section, what two primary factors determine the effectiveness of PNS axon regeneration, and how does the process of nerve regeneration occur in the PNS? And with that, we'll come back for section 12.6 on the plasma membrane of neurons.